By the way, is it okay to record your presentation, Sinem, today?
Yeah, that should be fine.
Sure. Yeah, it's 3 o'clock. I think we will shortly start, but as people are joining, I think let's wait for maybe another minute. So we will be starting in a minute. I think we can get started. Hi, everyone. I think today we are excited to have Dr. Sinem Saka from Harvard. And first, let me introduce her to you guys. Sinem Saka completed her undergraduate degree in molecular biology and genetics at the Middle East Technical University in Ankara, Turkey in 2008. She earned her PhD in molecular biology in 2013 from the University of Göttingen, Germany, where she was a Boehringer Ingelheim Fond's PhD fellow in the International Max Planck Research School, investigating protein organization in cellular membranes. She is a currently Human Frontier Science Program and previous EMBO postdoctoral fellow at Harvard Medical School and the Wyss Institute for Bio-Inspired Engineering at Harvard University, Boston. Her present research focus is to develop highly multiplex labeling and super resolution imaging techniques to investigate the nanoscale organization in cells. And with this, I think I'll turn over to Sinem, and please.
Thank you, Ahmet. Let me see if the screen sharing is working.
I'll stop.
Fine.
Mm-hmm.
Okay. Do you see the presentation at the moment? Like yes. The full screen presentation? Okay, great.
Yes. Mm-hmm.
Perfect. So, so thank you again for the chance to be here. Today I will talk a little bit about our recent work, but also the general context of repurposing DNA for microscopy applications. As Ahmet mentioned, oops, sorry. So far the transition is not happening, but let's see. Oh yeah, I think, okay, so we got it. So yes, as Ahmet mentioned, I've done my PhD in Germany in the University of Göttingen Max Planck Research School with Silvia Rizzoli, where I was focusing on organization of proteins in the plasma membrane using super resolution microscopy, mainly STAT microscopy. I've also developed some tools to correlate STAT and confocal microscopy with nanoscale and mass spectrometry imaging, so nanosims. And we have been using them to see the turnover of organelles in cells. I was really needing to do multiplex imaging because my previous work on protein organization really made me more interested in that to understand the general concept of the spatial organization for the nanoscale organization. So I went to Pangin's lab in Harvard University Wyss Institute, where they were utilizing DNA origami and DNA nanotechnology for imaging and other types of applications. So here I have been focusing early on utilizing single molecule imaging based on DNA paint, which I'll briefly talk about, and more recently using a SABER new technology for multiplex signal amplification. I think from that bio, it is clear that imaging has been one of my favorite research tools. And it is great because it gives us really pretty pictures, but beyond that, it also provides single cell, single organelle, single molecular information in the cellular and subcellular morphology within the context of organizational complexity of cells. So you can see the position effects, spatial context, and neighbors. However, there are also some limitations with imaging. So one of the first problems is the diffraction limit. So we end up with lower resolution with conventional fluorescence methods. Second one is a spectral overlap, which limits the multiplexing capacity. And third one, especially relevant for tissues, is the autofluorescence and scattering type of problems and the dynamic range of markers we are looking at, which make it harder to visualize everything with high sensitivity, and also decreasing the throughput we can acquire images because we need higher exposure times. So, So based on these, we actually need quite a bit of new tools and techniques to overcome these problems and bring imaging to where it can be really utilized as a power tool. And beyond these, there's also some more trouble when it comes to protein imaging, because proteins are very important as they are closest to the phenotype and function of the cells, but they are not easily sequenceable like RNA or DNA. So we need more methods that require affinity tagging, especially for the spatial part of things. So for these, common tools would be antibodies, nanobodies that are based on affinity interactions, but especially in the context of antibodies, which are by far the most common tools. We also have another issue that comes from the species overlap in the context of indirect IF, immunofluorescence. We cannot use, we don't have antibodies from many species, so that has an additional multiplexing issue when you want to use many antibodies. And for tissues, we discussed about this, the proteins are especially subject to these type of detection sensitivity problems because they have a high dynamic range. So some cells could have one particular protein at just like a few copies, whereas other proteins could be, millions of copies. So it's hard to really have the same method work for different types of expression levels and do that in the multiplex context. So there are like additional problems. For the detection sensitivity, there has been immunohistochemistry tools that contain, on top of secondary antibody amplification, enzymatic amplification using peroxidase and alkaline phosphatases so that they can generate colored products. But these, and really give high levels of signal amplification, but these are typically single plex, so it's not very easy to tune this amplification and also apply it in the multiplex context. So these are the current limitations. And in the context of multiplex protein imaging, there has been really many methods developed in the recent years. So this field has been really moving forward. There are scanning methods that utilize either metal or lentinide labeling or imaging mass spectrometry, as well as some near-infrared dyes and fluorescence dye combinations. These methods typically require special equipment and have low scanning speeds, but are very powerful. We also have spectral imaging methods. For example, the Sinem signal amplification system has been done in a sequential manner using the opal fluorophores, or Harper spectral imaging methods have been developed. These methods are also very powerful to be able to image in a really multispectral fashion and get high multiplexing. But the limiting multiplexing is still limited by the scope setup or the linear unmixing. And the signal discrepancies could be a big problem in this kind of settings, or they might really require special equipment to avoid that. One of the more established methods is cyclic labeling, or where you can remove or bleach antibodies and do relabeling every round. So there are already a few methods that go into this direction, and I think that's getting like really established at the moment. These methods are also very powerful, but they typically require long staining times because the antibody incubation is the slowest step, and if you especially have thick tissue samples, the steps take quite a while in each iteration. And the staining order also becomes an important parameter because you apply them one at a time, so you don't know if there is an additional bias coming from that. And more recently, the DNA barcoding has been gaining popularity. Again, this is a cyclic imaging, but at least the antibody labeling is done all at the same time. So in the context of earlier methods like exchange paint, universal paint, and more recently DNA exchange imaging and codex, the advantage here is that these methods are typically compatible with standard setups, although some fluidics might be necessary. And particularly, they are fast because you do one-step antibody incubation and then the order is not that critical anymore. So this has been the domain we have been focusing for a while as our lab on. So in my postdoctoral work, and I'll be mainly focusing on that So our lab started working with DNA barcoding in the context of proteins for super resolution imaging, firstly. So in this case, we are really utilizing the transient interaction of two DNA strands that are complementary to each other. So you can use the labeling strand that's called a docking strand, which gets bound transiently by an imager strand that has that fluorophore on. So each time these two hybridizes, you can actually create, localize signal based on the fluorophore, and then when they dehybridize, the fluorescent signal is like diffused in the background, so it's not really detectable anymore. So this gives us this kind of binding and dissociation over time, these events, and these binding events make it possible to actually do super resolution construction. So in the context of DNA origami, one could essentially really put docking strands in particular locations that you know how distant they are, so you can actually see the super resolution effect, and then collect many images where at low concentration of these probes, so you can find out, localize each of these spots and resolve individual DNA binding events. The spots can now be fitted so that each position of the spot is like very high, is obtained by high precision. And then by iterating these around thousands of frames, each of these spots can be detected and the images reconstructed. So in this case, we are looking at a 20 nanometer grid structure. So this was initially demonstrated on DNA origami structures, but it can also be done to cells, like partially or at the whole cell level. So we have been using a spinning disk confocal microscopy system for that, and it's also possible to use like a high-low system where the light is highly inclined. And this could be applied to proteins where by conjugating DNA sequences directly onto antibodies. So this is an early application where microtubules and TOM20 and HSP60 proteins were visualized through the whole thickness of the cell using confocal microscopy. Again, utilizing this principle, actually multiple colors can also be imaged with spectral imaging. And we can actually see here 3 different colors for the macro H2A protein and laminin B and nuclear phosphine. And it can be combined also with fluorescence hybridization, as we see here. So we have been continuing with these efforts in the context of super resolution microscopy with my colleague Yoshi Sasaki. We are labeling different elements of the nucleus and trying to see the localization of these different elements, understanding how they contribute to the organization of nucleus. Here we are looking at DNA, or ribosomal RNA, and fibrillarian, for example, that is really lining up the nucleolar structures. So, but of course, I think the main power of DNA comes from its barcoding capacity beyond this type of interaction control between the docking and imager strands. So we have been utilizing this principle also for multiplexing. The idea is, of course, very simple, as you might already know. We are separating the labeling and detection via DNA probes, where we now have the barcode oligo. And essentially, by changing the characteristics of these barcode imager fluorescent oligo interaction, one could determine how long this interaction is supposed to be. In the context of DNA paint, as I was talking before, we make this interaction very short-lived, but we can also make it by using longer strands more long-lived and do diffraction limited imaging or use different imaging setups so that this interaction is really lasting long and giving us enough signal for different methodologies. This also allows us multiplex the images by using several different orthogonal sequences, because although this binding is now more stable, it can still be reversible by changing the conditions in the buffer. So we could actually label many different barcodes like this in a cyclic manner. We have been utilizing this first on the context of indirect immunofluorescence, where we combine primary antibody with secondary antibodies, having fluorescent, instead of fluorophores, having DNA oligos for the tissue imaging in a diffraction limited setting. So in this case, you apply all your antibodies at the same time, and then image either one color or multiple spectral channels at the same time. And then after every round of imaging, exchange the fluorophores by removing the imager strand and adding a new one. So this could be done through manipulating the conditions in the buffer, especially the salt concentration. Or one could also do temperature or other additives like DMSO or pharmamine. So this was applied in the context of neuronal cells, and different markers were labeled by my colleague Yu Wang. But again, we were using mainly secondary antibodies for the concern of not having enough signal when we label with primary antibodies directly, since the element of signal amplification is gone. I mean, it's in principle possible to get enough signal, but we realize that there is a big utility for higher signal in the context of especially thick tissue images or FFPE type of samples, where the antigenicity is partially lost and the signal is not great for proteins. So we have been recently more focusing on developing a signal amplification system that can be multiplexed as well. So for this case, we are again using the same system and similar principles, but now instead of having one docking site, we are actually creating a concatemer of docking sites where the same imager can bind in multitudes. So this really concentrates the signal on the target molecule. And the way we generate this concatemer is based on a reaction that was developed in our lab called primer exchange reaction. So in this case, we have a primer, so a 9 to 10 nucleotide short oligonucleotide sequence that's single-stranded that can bind to a hairpin molecule. This is in vitro. So the hairpin molecule has this extension that we call the toehold site, where the primer can bind to complementarity. And once it binds, then a DNA polymerase can extend it further by displacing this complementary strand on this hairpin molecule until it reaches that stopper sequence. Afterwards, it gets automatically, spontaneously released due to branch migration events. And then this sequence happens over and over isothermally. So this, this primer is now extended, appended basically 1 unit each time. Here, the red part is what we call a stopper. This is where the polymerase cannot really move beyond, which means that when the polymerase reaches here, it's actually not able to leave, and that's actually promotes the branch migration process due to the stalling, and then it helps to release this concatemer every round. The stopper can be as easy as like just having a C nucleotide where we are missing a complementary G nucleotide. So in this case, we are utilizing a 3-letter code in the concatemer, which has the additional advantage of having largely, a little secondary structure because of missing the GC pairing. And that keeps the concatemer also quite linear. So this is the basic reaction we utilize to create long concatemers of single-stranded DNA. And because it reminded us of this existing signal amplification method, we are calling it SABER for signal amplification by exchange reaction. So it looks like a nano lightsaber. So one of the important properties of SABER is that the amplification is highly controllable. This process where we generate the concatemer, it's really subject to certain parameters. So by changing the hairpin concentration, the polymerase concentration, or magnesium concentration, or the length of the reaction, one could really modulate the length of these concatemers. And because they are generated in vitro prior to actually being utilized for stainings, you could also run them through the gel and see what length of concatemers you're getting. So you know what kind of amplification you're expecting to get based on this QC process. To enable high multiplexing, we computationally designed 50 orthogonal SABER amplifiers that based on criteria of orthogonality and then single-strandedness. So we tested 32 of these sequences so far in vitro for their extension capability. So like only one of them really failed. We can really extend them to the level of 500 to 600 bases with high efficiency. And then we also performed in situ orthogonality validations for these 32 sequences. One of these showed some crosstalk, but majority of them were actually really fine to use altogether. These experiments we've done by multi-well plate assays, where we applied either the concatemer with its cognate imager or the concatemer with all non-cognate imagers where we expect no signal, like in here. The case where we had a non-cognate imager giving signal was like, very little signal, but still a little bit of crosstalk. So we can eliminate that from the multiplex panels for the future. So the way that we utilize this for antibody staining is similar to the way we utilized it before. We actually have a DNA tagged antibody. Now having a bridge sequence, which would be utilized for immobilization of this PR generated concatemer, which has the binding sites, repeated binding sites for the fluorescent imager that is used for signal amplification. And both the bridge sequence and these concatenamers and imagers are designed orthogonally to allow multiplexing. So one of the first concerns for this method was, well, are these long concatenamers penetrating well into the tissue? So to test that, we have actually used whole mount retina preparations to see if our probes, antibodies, as well as the concatenamers are able to go into the cells and this kind of thick tissue preparations. And we could see that actually, this is about up to 100 microns deep for collagen and biomensing signal, that we have good penetration of these two markers, these two probes sets without problem. And then we can actually see here the overlay image in the flattened retina preparation. So we were able to see that in up to 100 microns, we actually have pretty good penetration of our probes, which was very reassuring. The second thing we wanted to check was, is the staining compact enough when we have these long probes? So that was another concern. Do we actually distort the imaging quality or the resolution for diffraction limited imaging? So here we are looking at microtubules because they are very densely labeled structures, and comparing the signal and the staining for the no amplification case, where we had an unextended primer that has only one binding site for the imager molecule, and then the SABER amplification, and immunofluorescence imaging for conventional IF imaging, where we use secondary antibody with fluorophore. So with this kind of amplification, we were able to get signal levels similar to the secondary antibody fluorophore, like comparable, and quite enhanced from the primary from the no amplification case. And we could also see that it's possible to see very nice microtubule structures, suggesting that we are still getting good labeling density. And when we do a full width of maximum comparison to see the resolution level, so in this case, we are using confocal imaging with 63x magnification, where 200 nanometer beads would be already beyond the diffraction limit. We are seeing that our immunoSABER is not giving us different resolution than secondary antibody or these diffraction limited beads. So it was at the diffraction limit, it's not making a big difference in terms of size of our spots. Next, we utilize it for the multiplexing in situ. So we are looking at 40 micron cryosections, fixed cryosections from mouse retina. So where we have labeled 10 different markers to utilize different cell types. Mouse retina has been a great tool for us because, at least for these validation experiments, because one could really see the different layers of cells. So it's good to really see any unspecific labeling and the specific signal from coming different cell layers. So this was a good demonstration of specific labeling of multiplex conditions. And another thing we have been actually trying to develop is to amplify the signal level further. This linear amplification we have been utilizing so far gives us signal comparable to the normal secondary antibody amplification, but just using the primaries. With the branching strategy, we are trying to take it to one level higher. So in this case, we are utilizing the primary concatemer as a binding site for secondary concatenamers that bind to these repeated units here, which creates an exponential signal amplification scheme. We have also checked that in the branch SABER case, where we now have a lot higher signal than secondary antibody fluorophore, we could still get diffraction limited resolution. So at this level, we are still not distorting our signal in the diffraction limit. We have been also pursuing this further to see if we can do even more iterations. So this is an example from human tonsil FFPE preparations, where we labeled the nucleus with DAPI, and then we have utilized Ki67, the proliferating cell marker, as our target. So we are looking at like a germinal center crop here. And as we go along, so this is these are serial sections. So we have the unamplified case, and then we have quantified the signal amplification for over this unamplified case, where we have done linear amplification with SABER with a 6-fold improvement. I'm showing the results with two different images with two different scales, so that we don't have saturation. So in one more round, we get actually about 50-fold improvement in the branch SABER, and we can do this iterative version where we do another round, use basically tertiary concatenamers, and this brings us up to like 180-fold amplification over the unamplified case, which would also help a lot with getting images a lot faster for large-scale tissues. So compared to the secondary antibody case, where we would get, for this instance, eightfold amplification, this makes a huge difference. And to do these quantifications, we have developed a unit-based nuclei identification by using manual annotation of contours to train the algorithm. So we can utilize really single cell level signal for the quantifications. And another thing we wanted to do was to see how it compares to some of the other amplification techniques commonly utilized for tissues. So one method that is especially popular is tyramine signal amplification. This is typically done on secondary antibodies where an antibody carries a horseradish peroxidase, and the tyramine molecule, which is typically bound to the fluorophore, in presence of hydrogen peroxide, gets activated and binds to the nearby tyrosine residues. So the signal is a bit dissipated around, but this creates really high amplification level. So on the same type of tissues, we have applied also tyramine signal amplification with using a commercial kit with different incubation conditions. And then we have done this quantification at the single cell level to see how signal changes for the whole tissue. So here we could actually see in a short or a long incubation of the tyramine molecule, it actually is very efficient signal amplification. But we could already see that even in 2.5 minute incubation, the signal level, the signal is actually starting to come out of nucleus. And Ki67 is a nuclear and also nucleoli enhanced marker. So it's actually starts as if you incubate longer, you get higher signal, but it also dissipates further from the target structure. In comparison, with SABER, we were able to still preserve the morphology quite well, even with iterative application where we done 3 rounds, or when we, for comparison, did secondary antibody staining with DNA conjugation with branching on top. So we can still see that we get nuclear and nucleolar labeling, and even we reach higher amplification or higher signal level compared to tyramine signal amplification. This is a big advantage, especially for automated segmentation type of methods, because for this kind of a setting, if you quantify nuclear signal, you would actually have mistakes because the signal is dissipating outside of the target structure. So we have also applied a combination of primary and linear and branch amplification together, so that creates some modulation based on the expression level of target proteins. In this case, we are looking at human FFPE tonsil sections. Again, this is a germinal sensitive area. We have 6 different proteins here. So CD8, CD3, and Ki67, IgA, IgM, and PD1. Among these, for example, IgA and IgM, these are again, secondary antibody type of stainings because they are labeling antibodies. So these are, although not found in too many cells, they are expressed in high abundance in the cells. They are expressed them. So we utilize only linear amplification for those, whereas for the other ones, we utilize branch amplification simultaneously to create this 6-color image and plus dotted staining for labeling different types of immune markers. So one advantage that comes with amplification, especially for tuning this level of amplification, is that you can now look at differently expressed proteins in the same sample without a huge change in your imaging parameters, and also do imaging fairly fast, which makes it quite applicable for high throughput imaging of big tissue areas like centimeter scale tissues. So in this case, for the linear amplified targets, we were able to go down to almost 1-2 millisecond level. And then for all of these 4 targets, we were able to actually keep the imaging times below 20 milliseconds per frame, which makes a difference when you have 300 frame tiles in a tissue section. So SABER is also compatible with amplification for in-situ fluorescence hybridization. In this case, the idea is quite similar, but you can actually directly extend the FISH probes via PER, primary exchange reaction, and then utilize these concatenamers for the fluorescence probe binding on your DNA, target DNA and RNA structure. This work, let me quickly move forward. This work has been largely driven by other postdocs in my lab, Josie Kishi, Brian Delivo, and then our collaborators from CONICET postLab, Silva and Lapam and MOS. And Brian, I think, is giving a seminar in a few weeks, so he will talk more about this. But they had some really nice examples on Stratina or for RNA labeling and on X chromosomes for DNA labeling, almost in 17 colors as well. So another application we have been looking into, is if we can also utilize SABER for expansion microscopy. The reasoning behind is that with expansion microscopy, the way it works is, so you have your sample, which is linked to a gel matrix, and then this allows you to digest unnecessary unnecessary molecules around if you wanted to create more space. And then in the hydrogel allows you to really expand the structure together with the gel, so that the points of interest are taken apart from each other, so there is more space. And then this really allows utilization of standard microscopes for super resolution imaging of tissues. This is a method developed by Ed Boyden's group. However, when you also expand the tissues, you're essentially diluting your fluorescence signal because there will be less fluorescence in a region. So we think that SABER actually might be really instrumental to do multiplexing in a fast manner and also with high sensitivity in expansion tissues. In this case, we stain with DNA conjugated antibodies, and then for amplification, we use acridide modified primers that can be linked to the gel, hydrogel, and then can be expanded with the gel. So in the proof of principle experiments, we applied it in cultured neurons for where we visualize pre and post synaptic markers, bassoon and Homer, and we can see that with expansion, we could actually get to this like high resolution level, diffraction beyond diffraction limit, where we can actually see these small diffraction structures separated. And this really helps in utilizing multiplex imaging because these expansion probes, samples are usually very thick due to the expansion process. So doing it in a cyclic manner, the antibody labeling takes really many, many days. But by doing the antibody staining, like one long step once, and then doing the the short fluorescent probe cycle in a cycles, that really helps to get high throughput because you can do the imaging a lot faster and in smaller time for more multiple channels. An additional advantage is that when you actually extend the tissues, you can actually separate these from each other. So you create more space as well that facilitates better binding of the probes by making the targets more accessible too. So this is the 6-color demonstration we have done on extended mouse retina sections as well. I'm sorry. Just a minute, I think it's having a problem with the transition. Okay, it seems it might have got stuck. Let me give it another second. If not, I will just stop sharing and probably restart. I'm sorry, it seems somehow the presentation has crashed. Ahmet, are you able to stop me from sharing? Because currently it seems I'm like
oh, you cannot. So
the computer
see.
maybe crashed, and I'm not. Oh yeah, maybe. Oh,
it works.
I think it's coming back. Let me see. Oh. Okay, now I'll just re-share. Somehow it got crashed, but
sure, no problem.
I think, let me see. Yes, we got it back. Okay, are you able to see that again?
Yes.
So, sorry for that. Currently we are trying to utilize SABER to scale it up for tissue mapping applications, because these type of applications really require multiplexing, sensitivity, and throughput, and we are hoping that we can provide these kind of demands and provide answers to these demands. So a few applications we are looking into, like we are using the human tonsils for our validation studies because it's relatively well characterized spatially. And then we are, we are also applying immunoSABER for kidney tissues in collaboration with Sanjay Jain, and brain tissues and gastrointestinal stromal tumors through collaborations as part of HubMap consortium and Human Cell Atlas consortium. So this is still work we are currently developing further, but multiplex imaging can be really useful to identify heterogeneities, especially in tumors. So together with Inga Marie Schaefer from Brigham Hospital, we are trying to use immunoSABER to identify key cell cycle abnormalities in GISTs, gastrointestinal stromal tumors. So here, high-risk and metastatic tumors have really genomic mutations that cause dysregulation of the cell cycle. And then using expression abnormalities of these key cell cycle markers simultaneously, we can actually understand how their functional interdependencies are and determine predictive and prognostic markers for therapeutic cell cycle restoration. So we have developed an antibody library that encompasses P16, RB1, phospho-RB1, P10, P23, PCNA, and AKT and phospho-AKT. So we also included phosphorylated markers here, which are sometimes problematic to see without amplification. And we are trying to understand how these are interdependent, both in tissue FFPE tissue samples from patients and also in cell cultures, patient-derived cell cultures, to understand what's happening also under drug response conditions. So this is work under progress. Hopefully we get to present some results and next time. So here we are in this project, we're looking at 20 different patient-derived samples from low-risk, intermediate-risk, and metastatic patients, and trying to treat different drug conditions as well to really understand what is happening at the cell cycle level. To summarize, we are working with these in silico designed primers, barcodes, and probes that we then use in vitro through the primer exchange reaction to prepare concatenants, which we utilize in situ for immunostaining as well as for fluorescent in situ hybridization. We hope that this modular system it helps to get the multiplexing easy and also scale up the multiplexing level. And I think one of the more distinguishing properties is the programmable amplification without in situ enzymatic reactions and independent tunability for different targets. And it's largely compatible with standard equipment and high resolution imaging, both in the context of diffraction limited imaging and also super resolution together with expansion microscopy. So we hope that can really make an improvement for high throughput biomarker discovery applications, digital pathology, and tissue mapping studies. And with that, I'll finish by thanking all the other people involved in the work. From my lab, Yu Wang and has been working together with me for the immunoSABER project, and Jocelyn Kishi, Brian Velivo has been leading the SABER fish project, and Brian is now in UW as well. So we'll get to see him in a few weeks, I think, and our collaborators from local institutions in Boston. And I'll be joining, starting next year, to European Molecular Biology Laboratories in Heidelberg Genome Biology Unit to apply some of these technologies to understand genome organization and also in different conditions of disease and health. So thank you for the opportunity. If there are any questions, I'd be happy to answer. And sorry for the technical issues.
Thank you, Sinem, for this great presentation. And we look forward to also your next great discoveries in the EMBL labs.
Thank you.
And sure. And today, I think let's make this more interactive because we have long time for Q&A. Why don't you guys unmute if you have any questions and then ask directly to Dr. Saka. If not, you can still type on chat. I'll be helping read them, but you may want to also unmute and ask too, to make this more interactive.
Sinem, this is Erdogan Memeli here, and I was wondering how quantitative can these data be through your multiplex method? In other words, are there ways to quantify the signals across of different proteins or nucleic acids?
So, I mean, I think it is quantitative in the fashion that if you use the same probe for the same target across different tissues, that would be quite quantitative. To compare the direct comparison for different protein targets that are labeled with different sequences, I don't think it will be very accurately quantitative because there is a bit of a difference from sequence to sequence. So in terms of the final amplification level, like the occupancy might be a little different from sequence to sequence in terms of how many fluorophores actually bind to these concatenamers. So it wouldn't be absolutely quantitative. I mean, I think it would be fine to make like low-high type of decisions, but if you want to really compare two different proteins that are labeled with different probes, that wouldn't be very quantitative. But I think already the immunostainings themselves, due to the antibody differences, are not super quantitative in the tissue context. So yeah, I mean, to be honest with that, it wouldn't be absolutely quantitative. But if you want to really compare conditions using the same set of probes, then it would be quantitative in that fashion.
Thanks.
Hi, this is Ben Grodner. I was wondering, with the amplified signal for each individual probe, any sort of off-target binding effects will also be amplified. So I was wondering, you have a certain increase in signal, but you also have a certain increase in noise. And I just wondered if you had analyzed the relative rates of those two, if you're increasing the signal to noise ratio, and if at a certain point you sort of have if you amplify more, you're amplifying the noise too much.
Yeah, so I mean, I think every amplification technique has that type of issue because as you amplify the signal, you would be amplifying the noise that you generate by your probes. So that is definitely, that's why we are really trying hard to optimize our conditions to decrease unspecific labeling from the beginning as much as possible. But I think at least for the RNA case, we were able to, utilize many different amplification rounds, I think 4 or 5. So it was still kind of reasonable. For the proteins, we have I tried to go beyond 3 rounds of amplification. And I think above that, actually it starts to, first of all, distort the signal level a little, distort the morphology a little bit because we are gaining, we are just becoming too big in terms of the probe size. And also, I think the noise amplification is getting too much as well. So we are not gaining so much more in signal to noise. I think up to the 3 iterations, it's still reasonable. And I think most of the time, probably 2 rounds of amplification is actually enough signal for most applications. So I think that is the level. But yes, there is definitely an inherent problem with amplification of noise. So you are as good as your probes, essentially, because if your probes are binding, you would definitely be amplifying them too. Currently, we are working on some related methodologies to make that better. So we actually reduce the noise while amplifying the signal, but that's still early work in progress.
Have you noticed improvements with the gel embedding and clearing?
Yeah, I think clearing does help. We haven't done like a great comparison yet, but we plan to do so. But definitely, you actually reduce the essential binding sites for like unspecific binding sites. So when you do clearing, so that improves the background quite a bit as well. Yes.
Hi, this is.
Go ahead.
Hi, this is Alden Moss from UC Berkeley. Great talk. So actually, this kind of goes with the gel embedded portion. So in the case of your expansion microscopy experiments, were the concatenamers diffused into the sample after the gel was polymerized and cross-linked? And if so, have you thought about like maybe the diffusion and penetration, how that differs in the hydrogel matrix compared to in a standard tissue case?
So in the particular case here, we had the acridite groups on the primers, so on the concatenamers essentially. So we first penetrated them and then cross-linked them and then expanded the gel. So it was only the small fluorescent probes that were that were penetrating through the volume in the expansion case. It is possible to do it the other way around, although we haven't tried it so far. We plan to try it. In that case, you are helping by generating more space between the target sites, so probably better binding can be obtained. What you asked one more thing, I think.
Yeah, I guess it's just, have you thought about like if it might be difficult to diffuse these larger concatenamers into the gel matrix compared to the tissue?
It is. I mean, we have to do more experiments on that. We have tried so far, up to like 100 micron or so cleared sections for the, clarity cleared sections in preliminary experiments. So we actually get pretty good penetration of the concatenamers. I mean, I think they are starting to have problems at the level of antibodies. The barcoded antibodies also starting to have penetration problems. Also, one thing to note is that the concatenamer length is quite important for the penetration. So we have realized that beyond 600, 700 nucleotides, the signal actually starts to go down due to probably penetration and access issues. So we start capping our structures around 600 bases roughly. And if you're doing branching, then we see an additive effect. So then you cannot really use two very long branches. We usually have one long and one shorter branch, and we saw that that is more effective than using very long branches. Again, probably due to accessibility issues as well. So yeah, the length is an important parameter. I expect that, I think there is a few preliminary experiments we tried in the lab, although I don't have any data at hand. It seemed like it is important to use relatively shorter lengths for if you want to do penetration after expansion. And very long lengths actually had a bit of a problem that we have observed that, yeah.
Thank you.
Hi, Sinem. I wanted to follow up on two of the questions.
Hi, Atar.
Hi, yeah, nice to talk to you finally. I've been emailing you a lot, yeah.
Yes.
So I think there was a question about quantification, and I wanted to ask, maybe you have an idea about this or not, but I think something we're very interested in is reproducibility in the long run. So do you think it will be possible eventually, if the technologies are commercialized or otherwise, to make big lots of these antibodies and store them so that you can at least get similar quantification?
I think that would be great. I mean, that would be great to have big batches of antibodies, because when you conjugate your own probes, there might be some batch-to-batch effects, definitely. So I think that would be great. We are still looking into the best ways how to store them so they are like very stable over time. There are a few methods we are looking conditions we are looking at. So that would be, I think, one thing. And then another thing we are evaluating is how, do you have any storage related effects for the concatenamers? We produce them in bigger batches and actually store them frozen and then tow in smaller amounts. So we are also checking if that is, there is any reproducibility problems with that as well. So far we have been utilizing them in big batches, and the antibodies, we usually, conjugate at the level of 100 micrograms. But I think if it's commercialized or if there are commercial antibody providers, big batches would be helpful, definitely. Also, in terms of, some antibody loss and final product you get, that would be a lot better to purify, etc. as well.
Yeah, that all sounds really promising. It sounds like you haven't seen any real big issues there with storage. It's just a matter of continuing to test. And
yes. But we do see a bit of a difference from antibody to antibody. So some antibodies seem to be a bit more sensitive to storage. Some are very stable over time. So there is, I think, anytime you do multiplex antibody stainings, you have to account for quite a bit of variation from antibody to antibody. So that is the general problem for multiplex protein imaging.
Gotcha. And then for the, there was a question about background and sensitivity or amplification of background. You might have alluded to this. Are you guys looking into anything that relies on dual probe binding? And do you envision any challenges with technologies like that?
Yeah, we are looking at dual probe binding approaches. That's like, so we would have active reduction of the background. So far that seems to be promising, but it's still a bit early, so we have to do a bit more tests. But yes, that's something we're definitely looking into.
Cool, thank you. That's
Yeah.
super exciting.
Thanks.
Any questions?
I have another one. What is the kind of cost analysis of SABER amplification versus ordering full-length amplified probes?
So it is actually quite like, so because you can make this in bulk, I mean, there is like one-time cost of, let's say, getting fluorescent probes, like a hairpin probe, etc. But once you do the capital cost of getting them once, you actually get quite a lot of material. So you could actually, like, I think the cost goes significantly down. I don't remember the exact numbers, but we have, I think, a cost breakdown in the SABER fish paper supplement to refer back to. I think it's like a few cents per reaction in the end for the amplification product. The additional advantage is that also when you actually buy these buy these concatenamers, it is hard to get them longer than 200 nucleotides, especially if you utilize 3-letter code. The companies wouldn't really provide that all that much. Or otherwise, if they do, it might be quite expensive. So there is that additional advantage that here you have the flexibility by using PDR to really design the sequences and get them to the length you want, and do quality controls in your own way, because you would have plenty of material to check and try. So that would be the additional advantage. But in terms of costs, I think it's probably like on the order of cents per reaction after you do the capital, like buying the things once. And then I think the only thing that we have seen that for ordering the hairpins, we usually order with purification, or you can do your own purification, because the reaction is a bit sensitive. If they are not well purified, then you have truncated products. So that is the only thing that costs in that direction. And then the enzymes, which are, mech labs, I think, provide them at very high concentration and quite cheap. So we typically do like a 1 milliliter almost level of reaction, which is enough for a long time to do amplifications.
I got another question, if that's good. I'm curious, I guess, in terms of how this compares to, say, hybridization chain reaction. Is the main advantage that you can get more multiplexing with this, or why is it advantageous over using the hairpins to do the reaction in situ?
So HCR is also a very efficient method of signal amplification, and it has been around for a while. So it's also a really nice method. I think in terms of advantages, disadvantages, so the design, the HCR due to metastable hairpins is a little more tricky. And I mean, I think we are just getting to the level of maybe 12 hairpins or so. Before it was, I think on the order of 5 hairpins that they 5 probe sets that they had. So multiplexing was a little bit limited due to design considerations. So here, I think it's a lot easier to scale up because the design is simpler. Another advantage here is that because we do we generate the concatenamer in vitro in advance, we can really quality control and like check that if everything is working fine, what length we get. Whereas when you do it in situ, you don't have as much control over the amplification process. And it's, so here we can actually tune the amplification level in advance, essentially. There you have to really do it empirically, I think. So that would be, I think, the difference. So probably easier multiplexing and a bit more control on the concatenarization process would be the main advantages.
Excellent, thank you.
Any more questions? So I have, go ahead.
No, you go ahead, Ahmet.
Oh, okay. Oh, I was just gonna ask, is there any commercialization path to SABER?
Yeah, we had some interests in that, but it's hard to comment on that. But yeah, it is in principle licensed, and it's unclear how the process is going to go. But yeah, so hopefully one day.
my only remaining question was, what level of multiplexing do you think is feasible, and what are the main limitations? It might be hard to answer that because I think every probe is a little bit different, but
Yeah.
what are the main challenges, or what are the limitations on that?
So I think like basically the main limitation has been getting the antibodies working well after conjugation. At least that might be, that's like, I think, a general DNA conjugated antibody problem. So because there is quite a bit of difference between how antibodies, like how each antibody reacts, and then some antibodies start to have more background issues afterwards. So we are trying to be careful and validate each of one of these after conjugation and optimize the conditions. So that has been, I think, the biggest time consuming part. Once you have a panel, that is relatively simple. So I think that is, and then of course also making sure that the antibodies are working fine. So at least it seems that we don't have a significant crosstalk issue with our probes based on our in situ experiments. So we do have like about 30 probes that is validated for in situ amplification without crosstalk. So that is definitely promising. We are at the, I mean, for the SABER fish, we have utilized the same type of sequences for like 17 color at the moment. And then we are at the same level for proteins too. So we are slowly scaling it up. In principle, we have like probes already validated for like 30, but it's more getting the antibody DNA conjugates working very well and then building on top of that. So I would say that is the really bottleneck step at the moment. But yeah, I think we're aiming more in the range of 20 to 30. And it seems like 10 is relatively simple to do. And then
you think fundamentally 50 or 100 is impossible, or you think?
No, it's not impossible. It's just will take some time. In principle, I think I have already like 50 concatemer sequences that we designed. We haven't tested all of them yet, just due to time constraints. But in principle, one could also easily design more. So you could do a lot more. But it's more of validating each one of them works under conditions, and depending on the tissue type, things might be a little different, etc. So I don't want to say like you can do unlimited number, but it takes a bit of time and optimization, but it's possible.
No, I think that's super exciting. I think in the time scale of 3 to 5 years, if you could get to 50 or 100 markers, I think that's a really fun place to be.
And one more question from Dr. Sohee Lee, and I'll just read from the chat. What is the base number of floors when you say 6-fold or 8-fold amplification is good?
Sorry, I'll try to stop share so I can see the chat as well.
Yeah, what is the base number of floors when you say 6-fold?
Base number of floors. So we did not quantify the base number of floors, but as the baseline, we took the unamplified signal level. So we basically it's a 6-fold over the unamplified signal level. And by unamplified, we are using a case where the primer was not extended. So there was only one binding site for the imager, essentially. We have not, it's not based on this many floors, because it's like basically aggregated data from some 600,000 cells in a tissue section. But yeah.
Great. Any more questions? If not, let's thank. Oh, you have one more?
Yeah, if that's okay. Sorry, I'll be quick. So it's just kind of a follow-up on the last question about the 6 to 8-fold amplification. So I guess if you have like a 600 length concatemer, I would think you have more than 6 or 8-fold more fluorophores, right? Is there like some sort of maybe self-quenching or something else going on that would cause kind of diminished amplification from what you would expect?
We definitely have less than what we would expect theoretically. We are still trying to figure out what is the main factor. So far, we were not able to like really pinpoint, okay, this is what's happening, because I think it might be a little different for each of those sequences, etc. So we thought about self-quenching. It didn't seem it's the main cause. It seems that somehow the occupancy rate is lower than 100%, but we are still trying to improve that part as well. So I think if we can improve it to like a higher occupancy rate, it would also make the amplification a lot more efficient for even with linear. But yeah, that's we are definitely not at the 100% there. It could be I mean, it could be that 3-letter code, there is a little bit of a slippage and some sites are not accessible. Also because we have units of like, let's say A binding site, and then it's going AAA. And for stable binding, we're actually occupying 2 of these AA units. So if there is like, there might be an A unit left in between each time, which might also cause some lower occupancy. So we are looking into like alternative designs as well to make it higher. But yes, that is definitely the case that we are less than 100% occupancy.
Interesting, thank you.
And then final question. Yeah, if not, let's thank to Dr. Sinem Saka again for her great presentation. And with the hands, right? I think we should have a good Zoom effect for this. And next week, we will also host Dr. Silas Maniatis from New York Genome Center. And he'll be talking about spatial result multimodal analysis of neurodegeneration, I believe using spatial transcriptomics, the NGS-based spatial omics technology. And we hope to see you next week and have a nice weekend. Bye-bye.
Thank you very much.
Thank you.